Hello everyone, my name is Zeshan Shoaib and I am a research professor in Intelligent Neuroengineering Lab of Korea University, and today I am going to talk about an interesting article, Neuromodulatory Effects of High Density Transcranial Alternating Current Stimulation and Direct Current Stimulation on the Prefrontal Cortex. And this is a combined study with fNIRS and EEG devices experimented on resting state subjects. This article is published by Dr. Usman Ghafoor with corresponding author Professor Kim-Shik Hong from Pusan National University, and they published this article in 2022 in a journal, IEEE Journal of Biomedical and Health Informatics, whose impact factor is 5.77, and this is a Q1 journal in mathematical and computational biology category, having the rank within top 10%. This article will explain the effects of tACS and tDCS stimulation devices on prefrontal cortex of human brain. tACS and tDCS are the transcranial electrical stimulation techniques to stimulate the brain with a very low electric current between 1 to 2 milliampere, and tDCS is a well-known neuromodulation technique that produces changes in brain functionality, while tACS is an emerging technique to modulate the neural oscillations during mental activities. So the main objective of this study is to find the answer of the question that whether tACS can cause any changes to hemodynamic response or not. To examine the tDCS and tACS effects on prefrontal cortex, they did 15 experiments, and in this study they used a device which can record fNIRS and EEG data simultaneously, and after recording the data they pre-processed the data, and for fNIRS data they used a low pass filter with cutoff frequency 0.1 Hz, while for EEG data they used a band pass filter from 1 to 50 Hz. And for EEG data, they extracted the bands delta, theta, alpha, low beta, high beta, and gamma bands from the data and measured the power spectral densities of each band. Yeah. In this article, they also introduced another interesting index called the bandwise power distribution, and this index can be estimated by individually computing the power of each band and then dividing by the total power of the frequency range from 1 to 50 Hz. So this index is very easy to calculate and can be useful for researchers in future to show their research results. Here we can see the optode configuration used in this study. For fNIRS data acquisition, they used 2 detectors and 8 sources, which we can see by blue circles, and the numbers in the circles are representing the position of the EEG electrodes, and here we can see a plus sign which is the position of anode in this study, and these minus signs are representing the position of cathodes, and they used 4 cathodes in this study. So this is the experimental paradigm used in this study. The experimental paradigm consists of 3 phases. a 6-minute pre-stimulation duration followed by a 12 minutes stimulation period and a 12-minute post-stimulation period. So the total time of experiment was 30 minutes, and in a stimulation duration for both tDCS and tACS, they include 15 seconds for ramp up and 15 seconds for ramp down at the beginning and end of the stimulation. They also validate the results by checking the statistical significance of the groups. So firstly, they check the normality condition for fNIRS data and performed Wilcoxon signed rank test if the data not confirming the normality condition. Otherwise, they applied the paired t-test for fNIRS data. Means if the data was confirming the normality condition, they used paired t-test to measure the significance, statistical significance of the data. And for EEG statistical data analysis, they only applied a paired t-test to compare the mean band powers before and after the stimulation duration. And in all statistical analysis, a p-value of less than 0.05 was considered as a significant value. Here we can see the results they obtained in this study. In figure 2, they showed the effects of tDCS and tACS on oxy and deoxyhemoglobin concentration changes in 3 time windows: before the stimulation, during the stimulation, and after the stimulation duration. After the stimulation, we can see the significant increase in oxyhemoglobin and a decrease in deoxy concentration changes in both type of stimulation, tDCS and tACS as well. The upper figure is showing the effects of tDCS and lower figure is showing the effects of tACS. We can see a greater initial increase of HBO, oxyhemoglobin, due to tACS, but decrease and then increased gradually. In contrast, the tDCS response increased gradually from beginning and stabilized after the stimulation. And in figure 3, they showed the channel-wise behavior, like channels near to the anode and channels little far from the anode. For both stimulations, tDCS and tACS, we can see the high amplitudes of oxyhemoglobin for the channels near to the anode and less increase of oxyhemoglobin in the channels far from the anode. In this slide, we can see the results for EEG data. In figure 7, they showed the band-wise power comparison before and after the stimulation for both tDCS and tACS stimulation. In tACS, we can see a significant increase in alpha band power and low beta band powers, while in tDCS stimulation there is a visual increase in all band powers but not statistically significant. And if we see the band-wise power distribution graphs, there is a 15% increase in alpha band after the stimulation, and 9% decrease in delta band power for tACS stimulation. While in tDCS stimulation, all band power distributions are almost same. So in this study, we saw that they modulated the prefrontal cortex and exhibited an increase in the oxyhemoglobin response and a decrease in the deoxyhemoglobin response due to tDCS stimulation. The same response caused by tACS stimulation but was slightly lower in magnitude. This finding is different from the previous reported in fMRI literature, where the bold signal was not attenuated by alpha tACS during rest. This may be due to the areal difference of stimulation electrodes. Means in fMRI study they used the electrodes on the visual cortex, but in this study they directly used the electrodes on the prefrontal cortex. The application of tACS has also been reported to produce phosphine via retinal stimulation. This phenomena can be a distractor, thereby reducing attention and affecting the results of task-based studies. So, and this study also showed that fNIRS signals measure are unlikely to be affected significantly by transcranial alternating current stimulation artifacts, but not in the case of EEG. In conclusion, we can see that they discovered significant improvements in mean hemodynamic responses during and after stimulation periods caused by both tDCS and tACS. The between group comparison revealed significantly less mean hemodynamic responses caused by tACS compared with tDCS. They demonstrated the feasibility of a high density configuration in increasing the hemodynamics in both hemispheres simultaneously through not only tDCS but also with tACS. And an increase in alpha band power and a low beta band power was observed after the tACS stimulation. These findings suggest that both sides of a prefrontal cortex can be targeted and that tACS and tDCS stimulation techniques are equally effective in high density configurations, which may be of clinical relevance. So this is the end of today's presentation. I hope you enjoyed the presentation. Thank you.